So here we are. You may have noticed I have a whole bunch of corn here, and that's because just because I love corn, and I do, but very essential for learning about genetics. The whole idea of what are called transposons or jumping genes were discovered by Barbara McClintock. Just groundbreaking research, all from just breeding these corn plants and seeing how the colors changed. Excuse me, how they're able to jump from one plant to the next. Just real exciting how something so simple, but with a lot of careful data collection and a good experimental design, can really discover a lot. So this is chromosome 9, as you can see here. And remember, check out the shape of this chromosome. Normally chromosomes are shaped like this. We normally think of chromosomes looking like this. That's because we've been taught about mitosis and meiosis. And this is what it's going to look like right before mitosis. But list some genes and the diseases or the traits they're associated with. So maybe write down Tangier disease, maybe bladder cancer, ovarian cancer, sialuria. I don't know what that is. I don't know what most of this stuff is. This is pretty serious stuff. Tuberous sclerosis. I've heard of multiple sclerosis, but that's a new one. Joubert syndrome. So maybe do a little extra credit. Tell me what some of these are. I'd be interested. But you see there's 136 million base pairs, so there's 136 million of these. Okay, so pretty cool. We know mostly what's all there. And remember I told you in class how we thought humans are so special. We must have 100,000 genes. Well, as you can see here, as of last year, 2,412. So sad. We know from the book, other simpler organisms that we think anyway have more genes, like up in the 20, 60, 80,000 range. I think it's some worms that we saw. But yeah, these are all the chromosome numbers here, 1 through 23, and you have your X and your Y, the number of protein encoding genes. So you see chromosome 1, which is the biggest, it has the most at 2,000. Y chromosome, pretty puny, 71. You look at the X, 842. Think about that. Women have a lot more genes than men do. 780 or so for more genes. So think about that. I have more genes. So this is from our Recovering the Romanovs activity, which is one of my favorite. We have the female skeletons that were found in the grave compared with the Duke of Edinburgh. And remember, he's a maternal relative of the Tsarina and all of her daughters. So they should have the exact same mtDNA. And if you look at the sequence, we do see everything matches except skeleton 9 in every circumstance. This is the only letter that doesn't match. So we know all of them, numbers 3, 5, 6, 7, they're related, but not number 9. That must have been who? The nurse. That was the only other female found there. So ABO blood system. Some of you know your blood type. If you have this blood with A's all over, that means you're type A blood, which means you're going to go after B blood. This is non-self. So you're going to have, this is what I am, I'm A blood. So I have all these B's floating around ready to attack the B's. This right here, as you might guess, is B blood. It has A's ready to attack my blood if it ever got in their body. If you have A's and B's, means you're AB blood, which means you're not going to be attacking anything because that'd be like attacking yourself. None. And then you see O blood. This is an interesting thing. Make sure you understand O blood has No, none of these proteins, no A, no B. So it's definitely going to attack A and B. And since they don't have any, A, B don't have any antibodies, they're called the universal acceptor. All right, just had a bunch of popcorn. And O is the universal donor. Because they don't have any these blood proteins to mess people up, so they can donate it to everybody. Yeah, pretty cool. So they add, blood. O blood is very valuable, but so is A because there's so many A's, like 40%, 40% of people. Actually, it's a little bit more, maybe about 41%, 41 to 42% have these blood. So pretty common, right? About half, based on my math. It's popular enough that Red Cross kept calling me one year every 2 months, and I basically got anemia because they were taking too much of my blood. So this is a diagram to show us about sickle cell anemia or sickle cell disease. Sickle cell disease. Hey dog, what's happening? That's the pups. That's the pups. This is the pups. Hey dog. I don't have any food yet. I'll come get you some food. Okay? In a minute. So, normally it's a T at this codon. And that means it's an A. Gets translated into an A. It's codes for the amino acid GLU, glutamine. And what will happen is somebody's sickle cell disease will mutate. It will turn into an A all of a sudden. And that it would be a T if there's a DNA, but there's no T in RNA, right? So and this is RNA, so no T. So it's a U. GUA codes for valine. And valine is very different. That gets in the protein. That's a real problem for your body because, as we'll see in the next slide, thank goodness, this is showing you all the different amino acids and what their properties are. So glutamic acid right here is acidic, and it also happens to be polar. So polar acidic. It changes to something over here which is basic and nonpolar. Two very different. They're going to fold up much more differently in the cell, in your cytoplasm. So that's going to create a structural change that's going to cause big problems. So that's the, see how it goes from G. Now, if it went from glutamic acid to aspartic acid, look, they're both very similar chemically. It probably wouldn't cause a change. Or a tyrosine to a threonine, no problem. So these two here, if this changes, that's be no problem. Oh, come on, no problem. It's going to be no problem. Other thing I want to look at is you look at all these, you'll see they all have the same backbone. This central carbon here, and then the amino group and the carboxyl group. Central carbon, amino group, carboxyl. Central carbon, amino group, carboxyl. So why don't you just go ahead on your paper, just circle the R groups, on a few of them, not all of them. That might be a waste of time, but I just want to get a feel for, oh yeah, so this is how these are different. Remember, the central part's always the same. Carbon, hydrogen, R group. Example, this is the simplest one, alanine, just CH4 group. And then we have the carboxyl, the amino group. There we go. And so the central stuff, and then this is different. And we can see that. See central carbon, and then this is what's different. So this is, this is the genetic code that we talked about. before, which is pretty awesome. They came up with this, how they discovered it. How these codons code for all your different amino acids. There's 20 of them. And yeah, we see also, just take a note, look at leucine. It has 6 different codons. If you notice, well, this is a little different, but most of the leucines start with a CU. But the third one's different. The threonine, the first 2 are the same. Alanine, the first 2 are the same. So that's a pattern I want you to recognize there. First 3, same. So this is what sickle cell blood looks at. So sickle shaped, like the Grim Reaper, the scythe that it carries to cut things, be like people, I guess, for the Grim Reaper. That's that sickle. Use to cut wheat. That's was the old style way of cut wheat. So you have sharper edges, and you can imagine this would get stuck in the vessels, because you have to go through very tiny, teeny tiny vessels like that are only this thick, so they can get stuck. So people with sickle cell, they can have a lot of damage to their organs because those blood cells don't get through very well, and that causes a lot of pain, hospitalization. Dehydration is really important for damaging for somebody with sickle cell. It makes these cells flip into this more. So this is normal. You got RBCs, and check this out, a little different. Guess what it is? It's a WBC, a white blood cell. So remember, you have a lot more red blood cells compared to white blood cells. And here's somebody with sickle cell. You can write normal on that other slide. So this is sickle cell blood, again, with white blood cell, but see these different shapes, and these just aren't going to flow as well. Plus they die earlier, die faster. So somebody with sickle cell is going to have less oxygen to their tissues, which is going to make them feel tired. Okay. And we discussed this. Every species have a different number of chromosomes, and the pattern we see is no pattern, right? Even simple ones, tons of chromosomes. Complex ones, life forms like us, fewer than some of these others. Okay. In somatic, it just means body cells. So any type of your part of your body, the skin, your muscles, your eyeballs, your hair. Gametic means sperm and eggs. Okay. And here's just kind of cool, showing you all these different species and how many chromosomes. The silkworm has 56. We have 46, and on down the line. Fruit flies have 8. I think I told you. They had 6 the other day. So once you go on this database, and I made a little video, it'll come on next. I'll show you how to use it. So what you want to do is go to this website, bioservers.org. You can register. All that does is allows you to save stuff that you work with, and we'll work with it later on this year. Either way, log in, or you can enter. We want to be on sequence server. See how I did sequence server? And when you get to here, I have a whole bunch of things here, but yours will be empty. You want to click on manage groups, then up to classes. And then you can pick, say, any DNA, but I would look at like non-human DNA. Check that out. Ooh, and look at, you can select all these things here. So say, for example, maybe do primate and birds. Let's compare, like chimps and gorillas and us to birds. So here we got primate. We got chimpanzee, but we could try a gibbon, gorilla. Let's do a gorilla. And go for birds. Let's do a Peking duck. And let's uncheck everything here. Make sure you check off the two things you're comparing under compare. So primate and Peking duck, and you're going to do a clustral W, and then click compare once you have those two things selected. And here we have our comparison. You'll see the western lowland gorilla's letters are here, the duck's letters are here. Just ignore all those areas that have dashes. Those are areas that just did not sequence correctly. So focus on where you see letters here. So on your paper, what you do is write down the gorilla A and Peking duck G at nitrogen base, and you write the number. This goes 0 to 25. So I would do count back. So this 25, 24, 23, 22, 21, 20. So it nitrogen base 20, you have A for the gorilla, G for the duck. Then at what would be 21, T for the gorilla, C for the duck. Then I would skip to say this one here, and this would be 26. So 26, 27, 28, 29, 30, 31, 32, 33. So 34, at base 34, you have a C for the gorilla and G for the duck. And just move on like that. 34, that'd be 35, A for the gorilla, G for the duck. And do like 10 of these, just do like 10, because you see there's a whole bunch. But again, just look to see where you have, for example, here would be a good one. You have T here and a T and a C, those are different. This would be 176, 77, 78, 79, 80, 81, 82. So 182. T for gorilla, C for duck. All right, so hopefully that works for you. Okay, so if so what allele is a version of a gene. So for example, maybe the gene for eye color. So you have a gene that codes for eye color. You have different versions. Say you have the brown allele, you have the blue allele, you have the green, and there's probably a couple more. We don't really understand eye color too well. And to kind of think of this question, mutations that could be considered beneficial, think of what an eagle has to do. Fly well, hunt well, swoop down. So maybe better muscles for flight. Maybe, stronger. Maybe endurance. Whatever it might be, strong. Maybe, the coloring, the camouflage. So, being able to blend in with the background better for hunting. Maybe better eyesight. Better eyesight. Maybe the talons are sharper, longer. Maybe the beak has a more beneficial shape. Okay. And why? Oh, a little possum outside. Hello, possum. And why they come in pairs? Because of sexual reproduction. Now, why is that? Let me have you think about it for a second. In order for sex to happen, you need to have one of these and one of these, right? And for humans, they need to divide equally in two. So there needs to be equal number for even division. So when they divide, they're evenly divided, so they come back and make the same amount. That's it.